Okay, so now I will first introduce myself before proceeding to today's meeting. So, good evening to everyone who have joined the meeting till now. My name is Moshumi Deb. I am a PMRF fellow, PMRF research fellow in IIT, in the Department of Chemistry and Chemical Biology, IIT ISM Dhanbad. And this is the weekly problem solving sessions for the NPTEL course Organic Chemistry in Biology and Drug Development. This is the week 3 live session, and this live session we will cover from the week 3 course contents, which was from lecture 10 to lecture 14, and we will discuss some 10 to 12 questions based on today's this week's course lecture. These questions will help you to solve your 2 weeks assignment, which will have conceptually similar questions in your assignment, and at the end of the session I will take any confusions, any doubt at any point of today's session. If you have it, you can always write in the chat box, and I will answer those at the end of the today's session. And still, if you have any question, any doubts, you can always write in the discussion forum, or you can personally mail me. I will share my email ID shortly at the end of the session. So, let us wait for 2 minutes more before we start today's session. If any one of you have weak internet connection or any problem, don't worry, I will just upload the video of this session in the YouTube and share the link with you, and also these slides will also be shared to you, so that if you have any if you miss any case, any point of today's session, you can always check from that slide and that YouTube video. So, I think we will wait for just okay, somebody wants to join. Okay, so we will wait for 2 more minutes, and then we will start today's session. Again, I am saying, if you have any doubt at any point of today's lecture, please do write in the chat box. I will answer that at the end of the session, and if I cannot answer it today, I will definitely take a note of it, and I will discuss it at the beginning of next lecture. Yeah, I think we should start. If anybody joins, I will permit it. So, I hope my slides are visible. Just write in the chat box if it is visible, the slides. Again, I am asking, just any one of you write if the okay. Thank you so much. Now we will start today's session. Okay. So, the first question is, what are the primary stabilizing forces of alpha helix and or beta sheet structure? Now, alpha helix and beta sheet structure, these are called the secondary structure of protein. Now, what is the primary structure? These are called the secondary. Now, what is the primary structure of protein? Now, we know that protein is made of amino acids. When 2 amino acids are linked together, the peptide bonds are formed, and by this sequence we have a range of amino acids, and this chain, it is called the polypeptide chain, and it is the peptide chain, it is called the primary structure. Now, what is alpha helix and beta sheet? Now, this primary structure of the protein, this range of this peptide bonds, when they interact with each other. Now, we know that there is a carbonyl group present. What is the structure of the peptide bond? There is a carbonyl group, there is a NH2 group, and we also have many side chain, and these chains, they are comparable, they can actually interact with each other, and those interaction makes this polypeptide chains into different shapes of alpha helix and beta sheet. Now, what are the options we will see here? The options are A, hydrophobic interaction, option B, hydrogen bonding interaction, option C, covalent interaction, and option D is disulfide bond. Okay, somebody wants to join, so let's admit. Okay, now I am the options I will again repeat it. So, what will be the answer of this question? So, it is very easy basically. If you know the basics of peptide chain, then you also know that the answer will be hydrogen bonding interaction. Now, I will show you. So, like I was saying, this is a peptide chain. Okay, this is one amino acid, this is second amino acid, and this is what is called the peptide linkage. There is carbonyl group, this is oxygen, this is carbon, and it is nitrogen in blue. Now, if there is one one peptide chain here and another peptide chain here, so what is there is a possibility that this oxygen of this carbonyl group, it will interact with the hydrogen of the NH group, and their hydrogen bonding is forming. So, these hydrogen bonding basically molds the polypeptide chains into different structures of alpha helix and beta sheet. So, if it is a primary stabilizing force of these secondary structures of protein. So, now I will show you the structures. So, yeah, it is called the alpha helix structure because it is looking like a helix, and it is called a beta sheet structure because it is looking like a sheet. Okay. Yeah. So, what you can see here, so look at this structures. If it is not so much clear in the meeting, I will share the slides. You can see from that, or in internet you can always type this structures. You will have beautiful structures in 3D models also, when you can easily understand. So, if you carefully look, it is a polypeptide chain. Okay. So, we have a carbonyl group here. Here is a carbonyl group present, and we have a NH group here in this chain. Now, there is a hydrogen bonding here. Okay. So, like this, in every turn there are few hydrogen bonding, and this is molds this structure in alpha helix, and this beta sheet structure in upper this on this plane, this sheet, we have a carbonyl group here, and we have a NH2 group, NH group here, and there is a hydrogen bonding which basically folds this sheets, and we have, which is called a beta pleated structure. Why it is called a pleated structure? Because there is some folds here present. So, it is called beta sheet or beta pleated structure, both are the same. So, I hope I am clear from this question, and if at any point you have any doubt, please write on the chat box. I will answer those at the end of the session. So, let us move toward the next problem, which says that which one of the following is not a weak interaction. So, again I am saying whenever you are doing this kind of assignments or this kind of multiple choice questions, always read the question very carefully. I myself do very mistakes when there is some true false, or there is which is not, which is type of question. So, always read the question carefully. What is it is asking? It is asking for which is not a weak interaction. So, the interaction, if interaction is weak or strong, we can know from the energy, and if you are do not need to know the exact energy, but just little concept if you have, you can always easily answer this type of questions. So, the options are here is A, pi-pi stacking, B, hydrogen bonding interaction, C, salt bridge interaction, and D, disulfide bond. Somebody wants to join. Okay. So, yeah. So, I will read the answers once again. What the options are: pi-pi stacking, the hydrogen bonding interaction, salt bridge interaction, and disulfide bond. Now, first I will show you the answer, and then I will discuss each of these interactions, so that you are clear why it is the answer. Yeah. So the answer is disulfide bond. It is not a weak interaction. All the three interactions like pi-pi stacking, hydrogen bonding interaction and salt bridge interaction, these are weak interactions. Means it is very easy to break this kind of interaction. But the disulfide bond, it is not so much easy to break a disulfide bond. Now what do we mean by pi-pi stacking? So if there are two phenyl rings like that, so we know that we have the many aromatic amino acid like phenylalanine, like tryptophan, like tyrosine. They have this kind of aromatic chain, like this kind of benzene ring. Now there is the pi bonds. We know that there are pi bonds here at the on the top of the benzene ring. So like this, there are pi orbitals here present at every point, and there is also pi orbitals present in this kind of ring. Now these pi rings, they are capable of interacting with each other, these pi orbitals, and it is called the pi-pi stacking. Now what happens when there is a amino acid like tyrosine or tryptophan present, if they are close nearby, they will interact with their pi orbitals, and it is called the pi-pi stacking. Now the next comes here comes the next, which is the hydrogen bonding and salt bridge interaction. So here is an example of lysine here. So what is basically we know as salt bridge interaction? It is basically a electrostatic interaction. Means there are two different charges, one positive and one negative, and if they interact with each other, it is called a electrostatic interaction, and it is called the salt bridge interaction. Now we know that the protein has a amino acid has a carboxylic acid and an amine group, and they stay as a Zwitter ion, not as a native state. Now at a freeze, so these Zwitter ionic forms, they can easily capable of interacting with each other. Now if there is a side chain like glutamic acid, it has a acidic side chain, and lysine, it has a basic side chain. So what happens in solution, these side chains are they have partial charges, like this carboxylic end has a negative and the amine end has a positive, and if they interact with each other, it is called the electrostatic interaction. Similarly, the hydrogen bonding interaction, this hydrogen of this amine group can interact with this oxygen, and this is the hydrogen bonding interaction. And last but not the least is the disulfide bond, which is the strongest of all. So we know that there are sulfur containing amino acids, right? There are so there is methionine, there is cysteine, which are called the sulfur containing amino acids, and they have a thiol side chain. Now if that sulfur, they just interact with each other and form this bond, it is called the disulfide bridge or disulfide bond. Now this sulfur bond, it is a covalent bond, and it is very much stronger bond than these of interactions. It is easy to break this kind of interaction, but to break this disulfide bridge, we need to apply more energy. So it is called, it is the more stronger interaction than these two. So I hope the question is clear. Just you don't need to remember the exact energy of these interactions or any kind of, because that is variable, but just remember that the disulfide is the most stronger interaction than these two. Now moving toward the next question, it says that which amino acid is termed as a helix breaker. Now what is a helix? I have previous in the first question I mentioned that it is a secondary structure of a protein. Now what is the main primary stabilizing force of the secondary structure? As discussed in the first question, it is the hydrogen bonding interaction. Now why a amino acid will be termed as a helix breaker? So it is very helix breaker. So if there is no hydrogen bonding, then what will happen? The helix will not be stabilized, and we have to choose the amino acid which is not capable of forming a hydrogen bond when in the peptide chain. Now what will be the answer of this question? I will first show you. So the answer is proline. Proline is the amino acid which is termed as a helix breaker. Now this is the structure of proline. This proline, it is a very exceptional amino acid because it is only amino acid which has a 2 degree amine or imine group. I mean, there is nitrogen which has only one hydrogen. And now what happens? We know when the peptide bonds are formed, the peptide bonds are formed, that is the amide bond formation. Okay. So the amide bond formation, for that what happens, that if there is a NH2, and there is a COOH. Now what happens, the water is eliminated and the formation of NH and CO. Now there is only one hydrogen here present. Now if it forms the a peptide bond, there will be no hydrogen present in with this nitrogen, and that is why the proline will not be capable of forming the hydrogen bondings, which is the primary stabilizing source of this alpha helix. I will show you here in the structure. So now look at this. This is a alpha helix structure. What happens? The hydrogen attached with this nitrogen interacting with this carbonyl of this chain, and the helix is stabilized. Now if there is a, when proline is present, there is no hydrogen present with this nitrogen, and since there is no hydrogen presence, there is no possibility of forming the hydrogen bonds, which is the primary source of the stabilization in a alpha helix. So what will happen? The helix will be breaking. So that is why proline is called the helix breaker amino acids. So I hope I am clear with this question. If you have any doubt at any point of today's session, please write in the chat box. I will answer that at the end of the session, or I will answer it in the start of the next class. So moving toward the next question, it says that according to Ramachandran plot, okay, somebody wants to join. So okay. So moving toward the fourth question. So it says that according to Ramachandran plot, which region is most allowed one? Now this Ramachandran plot, it is basically plotted between two dihedral angles. Now what is a dihedral angle? A dihedral angle, I will write here, now dihedral angle is basically the angle between two planes. When there are two planes, it is and the angle between, if you can easily understand it, if you think of a book. When you are opening that book, there are two planes. This is one plane and this is another plane, and the angle between these two planes is called the dihedral angle. So Ramachandran plot is basically plotted with in the x axis there is one dihedral angle, and in the y axis we have another dihedral angle. So first I will show you the answer, then we will discuss it a little bit. So the red region of the Ramachandran plot, it is most allowed one. Now I will show you. Yeah, so this is the Ramachandran plot. So we have this phi here and the psi here. So what are these? Like we know that when the polypeptide chains are formed, so there is a sorry. So when there is a carbonyl group here, and there is a alpha carbon here with which we have a R and we have a H present, and then we have a nitrogen and a hydrogen here. So this plane, the carbonyl group, it is on at one plane, and this NH group, it is on the another plane, and between this linkage, the planes, there are some angle. There is one angle is called phi and one angle is called psi. Just you don't need to remember everything about it. Just remember one thing, that this Ramachandran angle, Ramachandran plot gives us the most allowed regions. So there is, you can say there are 4 regions here. This first region, it has a red area which is written as beta sheet. Now why it is the written as beta sheet means it is the most allowed formation. I mean this beta sheet formation is most allowed one, and there is right handed alpha helix, it is also forming the red region. Okay, and we have left handed alpha helix, which means that it is in the yellow region. Now yellow region, what red region means it is most allowed, what yellow region means it is mediocre. I mean it can form, but it is not the most allowed one, or it does not have the most stability like the red region. And the white region, it is not allowed at all. So you can see there are no structure of the protein present in the white region of Ramachandran plot. So basically you need to just remember that there are 4 region, white region, yellow and red in the Ramachandran plot, and it is plotted with 2 dihedral angles, and it gives us which region is more allowed and most allowed one. and it comes from the modeling of the protein amino acid chains. So, it is written here, the red regions correspond to conformations where there are no steric clashes, and that is, these are the allowed regions, namely the alpha helical and beta sheet conformations. The yellow areas show that the allowed regions, if slightly shorter van der Waals radii are used in the calculation, that is, the atoms are allowed to come little closer, and this brings out an additional region which corresponds to left handed alpha helix, which I mentioned here. So, just remember this Ramachandran plot corresponds to 4 region, white, red and yellow, of which the red region is the most allowed and the white region is not allowed at all, and the yellow region, it if slightly, I mean, it may happen, but it is not the most stabilized one. Okay. So, now I will move toward the next problem. Okay. Now, it says that which one of the following nuclei is NMR inactive? The options are 1 hydrogen, I mean proton, 13 C, 16 oxygen and 31 P. Now, these are the mass numbers of these atoms, and now I will first briefly discuss about what is NMR, and you do not need to, if this is not an organic chemistry course, so you do not need to know the details, but since it is says that organic chemistry in biology, since there is organic chemistry, you should know that what is NMR. Now, if you want to read the details about NMR, I will just suggest some books at the end of the session, because today's time will not permit me to discuss NMR in detail. So, just know that every atom, it has we know that there is a protons, there are neutrons present in the nucleus and electrons are present at the outside orbitals. Now, these all protons have some charge, and due to this charge they have some spin, and since this have spins, when they are moving, they generate a magnetic field, and because of that, since a particle with charge and charge and spin, it generates a magnetic field. Now, if we apply outside magnetic field, that magnetic field is capable of interacting with the magnetic field generated by the atom itself, and that is forms the basic of NMR. It is called nuclear magnetic resonance. I mean, we are just that NMR formation is nuclear magnetic resonance, and this nuclear magnetic resonance means the magnetic field generated by the nucleus, it is we just resonance means it is interacting with the outside magnetic field, and this technique is the most powerful in detecting the structure, elucidating the structure of organic molecules. The proton NMR and carbon NMRs are most important ones when discussing structure of organic molecule. And so, there are not all atoms in the nature presents are NMR active. There are some atoms which are NMR inactive, and this question wants to know which of the following is NMR inactive. Now, I will first show you the answer and then discuss how you can tell by looking at an atom if it is NMR active or not. Okay. So, this is the answer, it is the 16 oxygen, it is the NMR inactive atom. Now, you can thought that how can we know if by looking at an atom if you don't know the basic of the NMR. So, it is very easy because there is a chart, so I will show you. Yeah. So, NMR active and NMR inactive nuclei are basically determined, or we can know it from the mass number and the atomic number of the atom. Okay. So, the mass number, it is present here, it is basically the sum of the number of the protons and the neutrons of a nucleus, and the atomic number is it is the number of proton, which is here written in the Z. Now, there is a chart here. So, if we have odd atomic mass and odd atomic number, so like in proton, okay, the proton, we know the symbol of proton is this, there is only one, yeah, it is 1 and 1. So, it is a odd number. So, it is NMR active. So, what happens if there is odd combination, it will be NMR active, and if now 13 C, now 13 is the mass number of the carbon, and the atomic number of carbon, as we all know, it is 6. 6 means it is even, and the atomic mass, it is odd. So it is odd even combination, means it will also NMR active. Now in phosphorus, we know that it has a mass number of 31 and the atomic number is 15. So it is again the odd combination, means it will be NMR active. Now comes the oxygen. Now this oxygen, it has the mass number of 16 and the atomic number is 8. So what happens here is a even combination, and I will tell you why in the, okay, somebody wants to join. So in case of this even combination, the atom becomes NMR inactive, and so just remember there are more examples. Now we can say from this chart you can see that if there is a odd combination, it will have half integer spin. I mean the spin is you don't need to remember the details. I will just suggest you the books at the end of the session. So if anybody is interested knowing the details of NMR, you can easily know from that book. So the this is called the spin of the molecule. If it is 0, then it will be NMR inactive, like carbon like 12C, it is NMR inactive. The oxygen 16 oxygen, it is also NMR inactive because these have the spin of 0. So there is, if there is no resultant spin, suppose the atom does not have any resultant spin, so what will happen? It will not generate a magnetic field, and if it cannot generate a magnetic field, it cannot interact with the external magnetic field which we are applying. So what will happen, that if it cannot interact, we cannot know, there won't be any resonance, and we will not have any signal for that atom. So but if there is a half integer, any non-zero values of the spin, so what will happen, it will capable of generating a magnetic field, and it will interact with the outside magnetic field which we are applying, and we will have a signal which corresponds which will give us detail about that molecule. So actually the proton and the carbon NMRs are most frequently used in the determination in elucidation of the organic molecules, and fluorine also used, and phosphorus we can also use these molecules to know the structure of an unknown molecule. And these techniques are highly used in the biomolecules like proteins also, to exactly know how the structure is happening. So moving toward the next question, if you anyone have any question, please write in the chat box. I will try to answer it at the end of the session. Now the question says that which one of the following protein purification process is not based on the charge. Now again I am saying that when you are answering this kind of question, just read the question carefully what it is asking. It is asking that which is not based. I mean it is very easy to do mistake in this type of questions, like in true false kind of questions. So just read the question carefully. So it is asking that which of the process is not based on the charge. The options are SDS page, ion exchange chromatography, option C is native gel electrophoresis, and option D is isoelectric focusing. Now I will just show you the answer first, then we will discuss. So the answer is SDS page. Now I will just discuss SDS page a little bit. So SDS it stands for, it is basically a long chain of a molecule. It is a salt, actually a aliphatic chain, and it has a sulfonate group at the end, and sodium is the counter ion of this sulfonate anion, and SDS stand for sodium dodecyl sulfate. This dodecyl is a aliphatic chain, and so what happens, that proteins may or may not have an overall charge. So it's not like if like the protein will have a overall charge, it may have or may not have. Okay. So but there is no consistent charge by size relationship like we need. So we what we need to use this sodium dodecyl sulfate. So what happens, like it has a negative charge, like I said that it has a sulfonate group at this, so 3 minus group at the end, and sodium plus, it is a counter ion for that. So this imparts the charge of negative charge of SDS. So SDS has a negative charge and it binds the protein evenly, and keeps it linear, so that there is a direct charge or length relationship. I mean, the longer the chain, it will have more charge, and the shorter the chain, it will have less charge. And this also goes for the molecular weight. Longer the chain, it also have a very high, longer the chain, it has the higher molecular weight than the shorter chain. So we have a weight by length weight, or we can say that the length can also represent here the weight of the protein or the protein chain. So there is a direct relationship between the charge and length, and so any natural charge, it will get overpowered by the charge coating it gets from SDS. So if it does not matter if the protein has overall charge or not, after getting coated by SDS, it will have a charge which is consistent with its molecular weight or the length of the chain. Then we can use that charge to send it throughout a polyacrylamide gel mesh. Now this polyacrylamide, it is a polymer gel, and we will just send it through this gel. This page, it is stand for the polyacrylamide gel, and the SDS is the sodium dodecyl sulfate. Now what happens, longer the chain, it will get tangle up, and it will move very slowly from the mesh. So you can say there is a mesh, it is like just a filter kind of thing. So if the chains are longer, it will it will move more slowly, because it will bind to the mesh and it will travel more slowly. But if the chains are shorter, and it will easily just tackle the, it will can easily just pass through this kind of the empty spaces, and it will just more fast, more slowly. So what happened, like you can say there is a charge gradient, it is a negative end of this it is the negative end of this mesh, and it is the positive end. Now what happens, if the chain is longer, like the protein has higher molecular weight, it will just move very slowly, and it will be at the upside and at the negative, more closer to the negative end. And if it is has a very smaller, it will have less negative charge, and it will travel faster and move faster toward the positive change, and it will be here. Now, by this technique, we can purify a protein. We can just we can just separate those on the basis of the molecular weight, and this process does not does not depend on the native charge of the peptide chain, because we are using SDS for imparting extra charge. So this is the basic of the SDS chromatography, and these all other three, these three are depends, they are just on okay, one second. This other three, they are just depends on the. Okay, there is one question, can you please explain the NMR's nature of C12C? Okay, I will just move toward that question. So yeah, 12C, what happens here, you can now for carbon, we know what is the atomic number of the carbon. The carbon, it has an atomic number of 6, and it has a mass number of 12. So in this is this falls in the this range, I mean it has a even atomic number and even mass number. Okay, since both of these numbers are even, it will have overall spin of 0. Since it has the overall spin of 0, it will not have any overall what should I say, there will not be a overall magnetic field generated by 12C. So it will not capable of interacting with the with the external magnetic field, which we will using detecting the NMR spectra. And if so, this 12C, it is NMR inactive. But what happens in case of 13C, which is an isotope of carbon, it has a odd mass number. Okay, so since it has a odd mass number, it falls in this part. Okay, it has odd atomic mass and even atomic number. So it has a net spin which has it is half integer. Now how it is come, you don't need to know this at point. I will suggest the book, you can read it from here, the discuss, because today's time will not permit me to make you understand the whole detail of the NMR. So just remember that since it carbon 12, it has even atomic and even mass number, it is NMR inactive. Okay. So I hope this questions are clear. this SDS page and also the NMR problem. And if you have any doubt at the end of the session or at the beginning of the next session, I will again discuss this. So, moving toward the next question, it is called that it is said that the complete proteome of a cell can be separated by SDS page, ion exchange chromatography, combination of SDS page and isoelectric focusing and isoelectric focusing. So, I will show you the structure and then I will discuss the answer of this question. So, the answer is combination of SDS page and isoelectric focusing. Now, this combination of SDS page and isoelectric focusing, it is what it is called 2D gel electrophoresis. So, first what happens, we all know that the protein, the amino acid, it has a pi, isoelectric point, at which it does not have any charge. It has net charge of 0 at pi. Okay. So, now if the pH it is less than the pi, it will have a overall positive charge. Okay. And if we have the pH which is greater than pi, so it will have a negative charge. Okay, somebody wants to join, just keep it here. Okay, so I will now okay. So, what happens in 2D chromatography? Okay. So, the first what happens that if at the pi the net charge of the amino acid is 0. So, if the pH when the pH is equal to pi, then what will happen, the amino acid will not have any charge and it will be just neutral. So, okay. And if the pH is less than pi, it will have a net positive charge, and if the pH is greater than pi, it will have a net negative charge. Now, what happen in the 2D gel electrophoresis, like that if we have to take the mixture of the peptides here, and in the first dimension, what it is called, why it is called 2D, because we are using 2 dimensions here. So, the first dimension, the isoelectric focusing is done. So, it is the basic end. Okay, the basic end. Basic end means it has the higher pH range and it is the acidic range. Okay, it is the acidic side, means it is at the lower pH range. So, what happens, the amino acids which have the proteins or the amino acids which have the less pi, which have if the pi are at the basic side, it will be stuck here. Okay, and if at the pi if like lysine or like arginine, they have a pi like suppose it has pi of 8. So, when the pH is equal to 8, so it will stop somewhere like here. But if it has pi like 3, like in glutamic acid or aspartic acid, so it will be neutral at this space. Okay. So, by this isoelectric focusing on the by the basing disk of the pi, we can separate. Now, what happens, we have to just tilt it 90 degree. Okay. And now it is placed in the SDS polyacrylamide gel, and this I have just discussed the SDS polyacrylamide gel, how it is happening. Then on the when the things are separated by this isoelectric focusing, now then we can just use SDS to just completely just separate or purify of these things. And we know that this is the side of decreasing MR means the molecular weight. So, this is in the SDS page, this is the range where if the proteins, the chain, if it has less molecular weight, it will travel more faster and it will be somewhere like here. And if it has more higher, it is longer chain or it has higher molecular weight, they will be somewhere like here. So, by using two methods, we can completely separate this kind of gel. So, I know this techniques may be a little bit of problematic to understand at the first area. So, I am just discussing the question. If you have doubt, I will again say discuss it in the next class positively. Just read from the modules and attend the sir's class. Again, if you have a doubt, I will discuss it in the next class. Okay. So, moving toward the next question. So, now the question is A catalyst can decrease the reaction rate by increasing the activation energy, increase the reaction rate by decreasing the activation energy, increase the yield of the reaction, decrease the yield of the reaction. So, first I will show you the answer, then we will discuss what a catalyst and what it can do. Yeah, so answer is increase the reaction rate by decreasing the activation energy. Okay. So, what happens like, yeah. So, this is called a kinetic plot. I mean, if you can say the reaction rates, these are determined by this kind of plot, and this energy at the y axis there is a energy, and at the x axis there it is called the reaction coordinate, which basically gives the updates like how the reaction is happening, how it is going, and the reactants, it will have energy. So, it does not matter, like here the reactants have higher energy than the product. So, if we just here it is right. So, the products are somewhat this energy and the reactants are somewhat this energy, and that may be, and the otherwise that the reactants have less energy and the products have more energy. Sometimes it happens also. So, what happens, it is called the transition state. Now, for a reaction to happen, the reactants have to attain a certain amount of energy, which is called the activation energy. So, this activation energy, after attaining this activation energy, the reactants can just react and form the product. So, the amount of activation energy we need to apply for a reaction to happen determines that the reaction will be more feasible or not. Now, if there is high activation, one reaction with high activation energy is not that much more feasible than the reaction which is have the low activation energy. Now, what a catalyst do? A catalyst is a molecule which just lowers the activation energy of a reaction, which means that if a reaction it is happening, it needs like this purple one, it is the reaction, the rate of the reaction, this is the activation energy, this amount which is needed for a reaction if there is no catalyst, and if there is a catalyst, this amount of activation energy is needed. Okay. So, the catalyst what is does it increases the reaction rate by decreasing the activation energy, means it makes the reaction more feasible, and if the reaction without catalyst is not that much more feasible, and the catalyst is a molecule, it will not change its structure or anything at the end of the reaction, it will be as it is, and it does not actually affect on the yield, it just makes the reaction faster and slow. Okay. So, what the catalyst do? The catalyst is reaction increase the reaction rate by decreasing the activation energy of reaction, and the activation energy is the energy which is needed to be attained by the reactants to for a reaction to happen and for the product formation. So, it is very easy, and we know the enzymes, they are called actually biological catalysts because the enzymes do the same thing catalysts do. The catalyst actually facilitate a reaction in the reaction vessel, but our body is also kind of reaction vessel, and enzymes do the part as a catalyst in the our body. Okay. So, the enzymes are also catalyst, and they similarly work how a normal catalyst do. They also increase the reaction rate, the reactions of our happening in our body by decreasing the activation energy. So, now moving toward the next question. So, it says that an example of oxidoreductase enzyme is fumarase, lysozyme, lactate dehydrogenase, RNA polymerase. So, I will show you first the structure. Now, if you can say that there are so many enzymes, how can we remember each and every example of the reaction? So, you do not need to remember actually each and every, the names of the reaction of the enzymes, they also suggest what kind of enzymes are those. Okay. So, the answer of this question is lactate dehydrogenase. Now, what oxidoreductase does? So, yeah, oxidoreductase, it is a term which comes from the oxidation, there is oxidation and reduction. Okay. So, the oxidation reduction means what will happen if there is a reactant, there are two reactants, the one will be oxidized and one will be reduced. Okay. So, this oxidoreductase does the reaction by reducing one substrate and oxidizing other, and there are several oxidoreductase enzymes, like there is one is called oxygenase. Now what oxygenase do? It incorporates oxygen into organic substrate. So if there is a substrate and there is molecular oxygen, now the oxygen will be incorporated in the substrate by the help of the oxygenase. So there is the called peroxidases. So what peroxidase does? It actually reduction of hydrogen peroxide and hydroperoxides. Now I said that there will be one substrate which will be oxidized and one substrate it will be reduced. Okay. So in here the substrate which is getting reduced, it is H2O2, hydrogen peroxide, and the it will oxidize the other substrate and it will be reduce itself to reduce itself. Now the enzymes they will just have the, in the name they will have this term peroxidase or oxygenase, like methane monooxygenase, okay, like there are peroxidases. So these enzyme have in their name they have these terms which suggest that they are what kind of enzymes. Like reductases, they just catalyze the reduction of the one molecule. Dehydrogenases, they oxidize the substrate by transferring one or more hydride ions. Oxidases, oxygen as the hydrogen, or oxygen as the electron donor, and as the hydrogen or electron acceptor. Okay. And hydroxylases, it adds hydroxyl group in the substrate. So all these enzymes, they all are a part of the oxidoreductase enzyme family. Now this if in the options you can see that this term dehydrogenase means one or more hydride ion will be transferred in the reduction of the substrate, and this name itself suggests that it is part of the oxidoreductase enzyme. So this is some, if you look at few examples, it will be more clearer that the name of the enzyme itself suggests what kind of enzyme it is. Like here lactate dehydrogenase, it is the oxidoreductase enzyme. So again I am saying you do not need to remember, just try to remember few important enzymes which are used in our metabolism in our body. So there are few enzymes, just remember those few and it will be sufficient. Now moving toward the next problem, which is called, which statement is false regarding induced fit hypothesis. Now again I am saying, and while answering this question, be very careful about the what is it is asking. It is asking the false. It is very much I have seen that many people do this kind of mistakes. I myself do this kind of mistake, like true false kind of, or which is not kind of answers. So just be little careful while doing this kind of question. So it is saying what is the which statement is false regarding the induced fit hypothesis, and the options are as follows. A, substrate specificity range is high. Option B is the active site is rigid. Option C is more accepted model for enzyme substrate complex than lock and key, and option D is conformational change occurs in enzyme and substrate. So I will just again show the answer first, and then we will briefly discuss that induced fit hypothesis for the enzyme. So the false statement among these four is the active site is rigid. Now discussing the induced fit model, what happens that induced fit, induce means it is, if it may not be fit for the substrate, but it will induce itself so that the substrate can bind. Okay. So here we can see there is an enzyme. So the somebody wants to join right now and we need to admit them. Okay. So in the induced fit model we can see that yeah. So the enzyme, it has an active site which looks like this roundish shape. Okay. And we have a substrate which have this kind of square shape site which has to be bind to the enzyme. Now when the substrate it is little bit closer to this enzyme, the enzyme will modify its structure so that the active site looks exactly like the substrate. Now we can see that when the substrate is nearby to that enzyme, the enzyme's conformation, the site active site, it's mold itself and now it is squarish. Okay. At first it was this kind of roundish kind of structure, and now it is the squarish. Now what happens, the substrate can easily bind to that enzyme. and after that the products are formation. So, it is called induced fit model because initially our enzymes active site it was not fit for our substrate, but when the substrate is coming near the active site of this enzyme, the enzyme is molding its side chain. Okay. So, we know enzymes are not nothing but the structure, some proteins are present here, very amino acids are present here. So, it will just fold itself in such a way that the active site it can now fit the substrate, and the reaction can happen and the products are formation. So, in the lock and key theory, what we can there is one so all theory, we all know that lock and key theory. So, the lock and key theory, it says that the active site of enzyme it is rigid. Okay. The active site of enzyme it is rigid and only it is substrate specific. I mean, if the there is it is not it will not make itself capable of doing the reaction with respect to the substrate, it is rigid. So, it is not that much acceptable than the induced fit hypothesis. Okay. So, just remember this thing, and it is very easy. If you can read it from anywhere, you will understand it. If you do not understand it, I will again discuss it next day about this induced fit hypothesis. Okay. Now, moving toward the next question. Okay. It says that in Michaelis-Menten equation, at high substrate concentration, the rate of the reaction becomes A, second order with respect to substrate concentration, B, 0 order with respect to substrate concentration, C, first order with respect to substrate concentration, and D, 0 order with respect to enzyme concentration. I will first show you the answer again, and then we will discuss this Michaelis-Menten equation and this question. So, the answer is 0 order with respect to substrate concentration. Now, we all know what is the 0 order reaction. The 0 order reaction is such that the reaction will not depend on the concentration of the substrate. Okay. So, that is called the 0 order. I mean the reactants. Okay. The reactants. The order is 0 of a reaction when it does not depend on the reactants concentration. I mean the rate will be constant whatever the substrate reactant concentration, the rate will be constant. Okay. So, I will show you first the, so this plot is called the Michaelis-Menten plot. It is plotted. So, it is plotted between the substrate concentration and the velocity of the reaction. Velocity of the reaction means the speed at which the reaction is proceeding. So, as we can see that it is this kind of graph it is producing. So, this kind of graph it is producing. So, as the substrate concentration it increasing at this side. So, when the substrate concentration is low, so what happens? This is a first order, it is a exponential graph here. So, this exponential graph it suggests that the substrate concentration is low, means there are lot of enzymes are available. So, the reaction is first order in this area. Okay. Now, this is it is called sometimes it is called the mixed order zone, because it is there is a competition between the enzyme molecules at this phase. Okay. And this is called the 0 order. I mean, there all the enzymes are occupied with the substrate. When it will happen? It will happen when the substrate concentration is high, means there are more substrate than the enzyme, and every enzyme is saturated with the substrate. Now, what will happen? If we increase the substrate concentration, now the velocity of the reaction it will not increase, because it will be constant, and it is called the V max. Why it is happening? Because there is no free enzyme left. Because there is no free enzyme left, there will not be any reaction and the rate will be constant. This part is 0 order part. Okay. So, this option like 0 order with respect to enzyme concentration, it will be correct. Okay. So, in Michaelis-Menten equation, just remember that this part, when the substrate concentration is low, it will be first order, and this will be the 0 order when the substrate concentration is high. And if you do not remember, if you remember the Michaelis Menten equation also, from that equation you also can derive this kind of answers from that equation itself. Okay. So, I will suggest just remember this graph, it is very important graph for the enzyme kinetics, and we will use it in the next modules, in the next lectures. This graph will be used frequently, and the Michaelis Menten equation also. So, read it carefully, and if you do not understand it, I will just discuss it in the next class again. Okay. And you can write in the discussion forum, or in I will share my mail ID, you can write also there. I will suggest some books, or I will just answer the questions. So, the last question of today's session is, the Lineweaver-Bark plot is associated with straight line with a slope of minus km by Vmax, parabola with an intercept of 1 by Vmax, straight line with slope of km by Vmax, a parabola with intercept of 1 by Vmax, minus 1 by Vmax. So, I will show you the answer. So, the answer is straight line with the slope of km by Vmax. Now, I will show what is the, so this is called the Lineweaver-Bark plot. Now, I will show you some things. Yes. So, the Michaelis Menten, so first I will discuss it a little bit. So, this is the Michaelis Menten equation. The V equal to Vmax S, concentration of S divided by Km plus S, where Km is the Michaelis Menten equation, Michaelis Menten constant. And from this equation, you can see now, I previously in the previous question I said that when the substrate concentration is very much high. Okay. Now, so when the substrate concentration is high, then we can ignore this Km. Okay. When the substrate concentration it is high, we can ignore this Km, and we can write this as only as substrate concentration of S. When it is much greater, it is much less than this, we can just write this equal to concentration of S. Now, this concentration of S and it will be cancel out, and we will have V equal to Vmax, and means it will be 0 order reaction. And if you have like 0 order reaction, and otherwise it will be first order reaction. If Km is much larger, we can ignore this S in the denominator, and then this V will be dependent upon the concentration of X, and it will be first order reaction. So, in previous question I mentioned you that if you forget that how this is happening from the graph, you are not able to answer it, we can you can also derive it from this equation. Okay. Now, Lineweaver-Bark equation, it is nothing but it is a plot of 1 by V versus 1 by S. So, in the x axis we have 1 by S, 1 by substrate concentration, and in the y axis we have 1 by V, the 1 by V. Now, how you will remember this? What is just remember what is plotted in this equation. That Lineweaver plot, we are plotting 1 by V versus 1 by S. Now, rearranging this equation, you just do it like you can just reverse it, and you can write as 1 by V, and then what will happen? The Km by Vmax, it is a constant, and it is 1 by S and plus 1 by Vmax. So, comparing it with the equation y equal to mx plus c. So, 1 by Vmax, it is our intercept. This intercept is 1 by Vmax, and slope of this line is m, it is Km by Vmax. So, it is the slope or intercept of this Lineweaver-Bark equation. So, there is one more equation is called Hans-Wolf equation. So, in that equation S by V is plotted against the substrate concentration. If again you can compare it with y equal to like mx plus c. So, here in Hans-Wolf plot, the Hans-Wolf plot equation, the slope will be this and the intercept is this. So, it is very it is in at one glance it is may seem like how we will remember what is the slope and what is the intercept of each and every equation. You do not need to remember at all, just remember which is plotted at what end. Like in y axis, that is plotted 1 by V, and in the x axis the 1 by S is plotted in Lineweaver-Bark plot, and in the Hans-Wolf equation S by V is plotted against the substrate concentration, and then just rearrange from this kind of things in y equal to mx plus c kind of equation from this Michaelis Menten equation, and you can easily say which is the intercept and which is the which is the intercept and which is the your slope of the line. And this kind of plots generally they are these are happens as in a straight line, these plots. Okay. So that's it, this was the last question in the today's session. And okay, I hope I was able to I was able to make you understand about the questions and the problems of today's session. And if anybody of you have any question, you can just write in the chat box now, or if you have any question, we also can discuss in the next class. Okay. So I will just quickly suggest some books for the things, like for NMR. I am writing in the chat box. For NMR, there is a book called by Organic Spectroscopy by William Kemp. So you can read NMR from this book, or there is also a book, it is called Organic Spectroscopy by Pavia. Pavia is a bit advanced book, but it is more which is more well written. And for the kinetics, you can also use any of the physical chemistry books you can read. Okay, for the kinetics part, for the enzyme kinetics part. And if any one of you have any question for this session, you can write now in the chat box, and I am sharing my email ID also, so that if regarding today's session, if you have any doubt, just drop a email, I will try to answer the question. And you can also write in the discussion forum in the NPTEL's website. There is a discussion forum written as ask a question. So you can also ask a question about today's session in the discussion forum. I will definitely answer it at that forum. And if any one of you have missed anything about today's session, do not just okay, somebody has raised a hand. Okay, okay, I will now just unmute you. So yeah, so one by one you can unmute yourself and ask your questions. I can say there are several hands, so if you have any question, Andhra, Chaitanya, if you have any question, you can write in the chat box, or you can unmute yourself and say. Anyways, today's session, this course video and this PPT, it will be shared to you. Okay, if Subhash, do you have any question? These slides and this video will be shared to you, and so if you have missed anything in the class, you can also just see from there. And in next class, we will cover the next week's course lecture.
Madam, I missed some of the lectures previously.
Okay.
Is there any chance to get that lectures, madam?
Okay, the lectures regarding this problem solving session, right?
Yes, yes, madam.
Okay, so there is a Google sheet, it is shared to you through email. Just check, I think it was shared to you in the previous class, or it will be in the NPTEL site also. So in that sheet you have a YouTube link and the document file of this course. Okay, so there will be some
Okay, madam.
like this kind of YouTube links and this kind of video link and this kind of document links. So from that you can access the videos and the lecture slides. Okay, and it is in the NPTEL
Madam,
website. It is in the
please,
NPTEL
please,
website.
and please say your mail ID, ma'am.
Yeah, I have written it in the chat box. It is 21dr0110@chem.iitism.ac.in. So if you have any question, you can mail me. I will answer that mail. Okay. Did you get the email ID from the chat box?
Not clearly observing, madam.
In the chat box, it is written.
So, okay.
It is 21dr0110@chem.iitism.ac.in. In the chat box I have written. Anyways, in NPTEL website there is also option ask a question. You can pose your question there also, I will just answer that from that part also. Just mail a question, email your question, I will answer that. So I hope you don't have any question right now, or if you have, just we will now just conclude today's session, and I will discuss in the next session if you have any doubts regarding today's session or the emails. Okay. Okay. So thank you all for joining today's session, and I will see you next Friday at from 7 p.m. Okay, for the next session. Yeah. Thank you so much. Thank you.